All right. So this is our great pleasure to welcome today Stefan Linquist. Stefan is an associate professor at the University of Guelph in Canada. Stefan is a philosopher of biology. He also has a background in biology, and his work touches upon many different topics of interest, including ecology, genomics, and evolution. If I remember correctly, Stefan also worked on cultural evolution, and really, this is one of these philosophers with oh, what is that? I'm supposed to confirm the language here. I don't know exactly why. One second, please. All right. Sorry about that. So yeah, so I think this is really, Stefan is really someone we wanted to invite for a long time, and now is the day after different attempts. So really, Stefan, thank you so much for accepting our invitation. Today, Stefan will talk about genome level ecology, a novel approach to the functional analysis of junk DNA. And I'm very excited about this talk. Thank you, Stefan. The floor is yours.
Great. Thank you very much, Tomas. I really appreciate the invitation. You were asking earlier about the images on this initial slide. And for me, these represent three ways that a genomics researcher can choose to think about the genome. So on the left, at least on my left here, we have an image of some unicellular organism, and every component pretty much inside that organism is doing something functional. So you could think of that at the level of the genome as if every bit of the genome, more or less, has some functional role to play or other. In the middle, there's a different sort of picture where we're in a junkyard and there's apparently one functional vehicle up in front here that we're going to focus on. And in the background, there's just a kind of vast mess of junky vehicles, and we're not going to pay much attention to those. So that's another way in which we could think about the genome. And over here on the right, I think is a depiction of more of a genome ecological view, which I'll explain in the talk, where there's certainly a certain amount of functional entities, some junky entities, but also in our perspective are a bunch of local ecological processes that help us understand the kinds of patterns that we're seeing. So it's basically bringing an ecological approach in at the genomic level. Okay, so here's an outline of the talk. For much of it, I'm going to be talking about the concept of junk DNA over 4 historical snapshots. And that's partly because right now I'm working on a book for Cambridge Elements on junk DNA. And so this is a nice opportunity for me to cover in broad brushstrokes some of the work I'm doing there and get feedback on it. I'll briefly then talk about a methodological problem that I think plagues the discipline of genomics right now, I call it the problem of causal rule myopia. And then towards the end of the talk, I'll present some examples that I've collaborated on with my colleagues at University of Guelph, where we're applying an ecological approach. Okay, so 4 historical snapshots here. I'm going to be covering first 1972 when, well, these 2 papers presented at symposia by Susumu Ono, who's generally associated with coining the phrase junk DNA. So at the time, let me just go back a sec. So at the time, the prevailing view, as arguably it is today, was that picture that I depicted on the left-hand side of the first slide, where most of the genome is probably doing something functional for the organism. And Susumu was one of the first people to push back on that idea. He tried to invert the picture and say, no, in fact, approximately 90% of eukaryotic genomes are not functional for the organism. And he offered 2 general sorts of theoretical arguments for that claim. So one of them was an argument that had been around at this point for a couple of decades. This is Muller's mutational load argument. And also he appealed to variability in genome size. Okay, so let me just briefly talk about what those arguments are, because They're pretty important, I think. And they're arguments that reoccur like every time the junk DNA debate arises. So let me see if I can adjust my screen. Okay, I don't know if that makes a difference. Okay, so at the time, empirical estimates were that about 10 to the minus 5 deleterious mutations occur per genetic locus. By deleterious here, we mean organisms that severely impact the viability of an organism or kill it. So basically, it follows that anything above 10 to the 5 genetic loci in an organism represents what he called an unbearably heavy genetic load. So the idea here is if you just have too many genes structuring an organism, you become vulnerable to genetic mutations that can basically cause the organism to shut down. So the idea was there might be an upper bound to the amount of DNA that's functional in an organism. And when you compare it to the total amount of DNA that is there, it's only a small fraction, maybe 10%. So that was an argument that's been around for a long time. Secondly, he points out the genome of the lowly lungfish and salamanders can be 36 times greater than our own. So lowly here, I think, is meant to designate that these are not particularly phenotypically complex organisms. Perhaps we could debate about that, but the basic idea is there's no way it takes 36 times more DNA to construct a lungfish than it does a human being. So what is all of that extra DNA doing exactly? Well, one of the pieces of the puzzle for Ono was, well, he was interested in this question of how do genes with novel functions arise in the first place? And this is a sort of theoretical problem, in part because if you think that most genes are under strong purifying selection, that is, if you think that any little mutation that affects their function is going to be weeded out by natural selection, then it's kind of a curiosity as to how you get a novel gene that has several different new mutations in it. And so Ono came up with this potential mechanism of gene duplication and drift. So on this idea, occasionally there are duplication events within the genome where one functional gene basically creates a second copy of itself by a sort of accidental process. And then that second copy is now free to accumulate what he called forbidden mutations and eventually, sometimes, acquire a whole new functional role. But most of these experiments, as he called them, failed to result in a second functional gene. They just go on to accumulate these mutations. So he says they might be serving useful but negative function of spacing those genes which have succeeded. So this is kind of an interesting functional category that he allows for, where it's not in virtue of the sequence that these copied and gradually mutating genes are potentially functional. So sometimes they'll acquire a secondary function in virtue of their sequence, but sometimes they'll just kind of serve as a useful spacer. But he did also add that he didn't think that much of the genome even has that much of a role. Okay, we'll come back to that in a moment. So how did Ono draw the function junk distinction? Well, functional DNA for him was DNA that's under selection for some sequence-specific effect on the organismal phenotype. Okay, junk DNA is potentially under selection for non-some specific sequence-specific role. It might be selected as a spacer, or not under selection for any organismal effect at all. So it's kind of already a category with two very different sorts of things in it. At the end of this presentation, there was a bit of a discussion, and that discussion has been documented in the proceedings of the conference. And E.B. Ford had a comment that is pretty interesting. He said, "I think the word junk is a powerful word. The only thing I would seriously question is the assumption that just 10 units would be sufficient to act as a spacer." So they're having a debate here over that middle category of functionality, where it's not sequence-specific function, but spacers. And Ford is suggesting, hey, maybe most of the genome is functional in that sense, of being a spacer. And Ono thinks I have had what I think is a pretty interesting response to this. He appealed again to that argument, the mutational load argument. And he says, if we argue that a given spacer can change its base sequence any way it likes, but that a length of it has to be conserved rigidly, then deletions would become deleterious to spacer function. It follows that spacer sequences contribute to the mutational load. And for this very reason, we can't afford to keep too many spacers. Okay, so again, the idea, it's the mutational load argument bumped up to the level of spacers, right? If they're under strong purifying selection, then we're going to see a lot of constraint. And that also, I think, ties in with this variability argument. Why would there also be so much variability among genomes at the same time? So he was skeptical that there's strong selection even on spacers. Edmund Eunice had an interesting comment, which I think reflects the sort of biases that were prevalent then and arguably now. This is what I emphasized earlier. He says that DNA must have a functional value since nothing is known so widespread and universal in nature that is proven useless. This seems to be a very pervasive assumption in the field of genomics and perhaps elsewhere, that if something is really prevalent, it must be doing something important. There was kind of a funny response by Marco Ficcaro, and he points out, well, you know, a lot of us university professors are hanging around, but we're seen as rather meaningless to the majority of our students. So maybe there's some exceptions to that rule. Anyway, I want to just imagine for a moment, I'm just going to set aside the historical debate over the meaning of junk DNA and just imagine ourselves as research scientists interested in isolating parts of the genome that have some function in light of the mechanism that Ono was proposing, this gene duplication mechanism. So what we run into is a problem of gradual functional decay. Okay, so when a gene is duplicated and one copy starts to accumulate mutations, it retains partial functionality for a period. In fact, the main gene may get knocked out, and that secondary gene now becomes relied upon as a redundantly functional. And even after it loses its functionality, by here I mean its capacity to generate a protein, it could continue to retain hallmarks of functionality for much longer. It still might attract transcription factors, for example. So this leads to a sort of terminological problem and a methodological problem for us as these research scientists. So we're using this concept, is it junk or is it functional? But when you think about this nature of decay, this gradual nature of decay, it really doesn't make sense to draw a binary distinction. We should be asking, even though it's a very awkward sounding phrase, and I don't recommend it, you know, how junky is it? How far along the process of mutational accumulation is it? And could it be recovered? And is it likely to be recoverable in some other functional capacity? Those are the sorts of more interesting questions, I think. And Ono was aware of that, but instead, you know, the way that things go sometimes, I think we ended up with a much simpler binary distinction. And again, there's a methodological question of just how do you distinguish empirically varying degrees of junkiness? And, you know, we won't want to adopt that phrase perhaps, but all right, on to the next historical snapshot. So here's another place in the history of genomics where the function junk distinction became a matter of considerable debate. It was around the publication of these two papers back to back in Nature, in the journal Nature, arguing, basically bringing attention to the role of mobile genetic elements or transposons in structuring genomes and what that meant for the search for genomic functions. So one of the main points being identified in these papers is that there are these elements in all genomes, eukaryotic and prokaryotic genomes, that are capable of copying themselves and then reinserting into new regions of the genome. It happens in not just somatic cells, but germline cells also. And so why is this important? Why is this theoretically significant? Well, what this means is that there can be there's a mechanism now where there's a, well, I guess for Doolittle and Sapienza in particular, they're pointing out a lower level of selection occurring within the genome. So you could have competition among transposable elements for how efficient they are and how good they are at inserting and finding regions of the genome to insert into. And so, as they say here, cells themselves are environments in which DNA sequences can replicate, mutate, and evolve. I just want to take a moment here to talk about transposable elements because it's kind of important, and I'm assuming that some people may not know that much about them because I certainly didn't until I started getting involved in this work. So here's, an anatomy of a transposon, and they've got these repeated sequences at either end. But the thing I just want to point out here is that they also contain their own genes, okay? They're open reading frames. So what these genes within the transposon do is they encode these proteins that facilitate them being basically snipped and reinserted into new regions. So they have their own proteins that do part of the work for them. Here's another diagram that I'll try to explain. So here's an example of a transposon jumping into a new region, and that can often have mutagenic effects. Okay, what I mean by that is it can disrupt the functioning of some gene that might be important for the organism. So it's often thought to be harmful for an organism to have a lot of transposon activity in its genome, right? These little elements are jumping around. They can land in the middle of a gene and disrupt it from being transcribed. They can get into the regulatory region and screw with that. So organisms have evolved various mechanisms for detecting transposons and silencing them. And one of the ways they do that is to apply methylation marks around the transposable element. Okay, so here's an example of some methylation that's happening around the transposon, epigenetic repression. And this is an important point, when that happens, there can often be some portion of the transposable element, some section of those genes that it has, that don't get methylated and that can be transcribed into an RNA. Okay, that's going to just happen as a consequence of this process of there's this kind of evolutionary arms race happening, and a consequence of it is there's going to be some kind of leftover transposon that gets transcribed, and you end up with these RNAs floating around inside the cell. Okay, so what sorts of challenges are associated with this? So transposons have sequence-specific functions, because they contain open reading frames, and they also can be selected at different levels. So we need a different notion of functional DNA now. That definition that Ono presented is not going to quite work anymore. So now functional DNA is under selection for a sequence-specific function at the organism level, but the junk DNA category is now getting expanded. So now we have DNA that's under selection for some sequence-specific function at the transposon level, right? So before, if you had a sequence-specific function, you were functional. Now, you can have a sequence-specific function, but if it's just for a transposon, then it's not functional anymore. It's junk, or it's selfish, sorry, which is a type of junk. Or you could be under non-sequence-specific selection, like a spacer, or you could have no effect on fitness. So at this stage of the discussion of junk DNA, you can see this category is becoming an increasingly large basket for various sorts of things, right? Some of which have sequence-specific functions, some non-sequence-specific functions, some which just aren't functional at all, okay? And one of the questions that we can ask is, what kind of work is this distinction doing at this point? I'm not going to get into that right now, but it's an important question, I think. All right, so next, I just wanted to highlight the fact that there was a lot of controversy surrounding those two papers that appeared in Nature. And for some reason, I still don't understand, there were like all of these other labels that were applied to this like non-coding DNA, polite DNA, ignorant DNA, and so on. And but the general theme of the criticism was, I would interpret it this way as, look, you haven't really proven that much of this DNA isn't functional for the organism, and shouldn't we, and here are some reasons why it might be sort of cryptically functional. So people were unwilling to let go of this idea that Eunice had described 10 years earlier, that, look, if it's there, it's got to be doing something good for the organism. Okay, again, imagine ourselves as research scientists, right? We're looking for the organism-relevant functional DNA. And now we find out there are these transposable elements. Well, the problem of gradual functional decay gets even worse. As I just a moment ago, I mentioned that there are these little transcripts that get generated even after the transposon has been shut down, right? And so it remains biochemically active. So this is a source of what you might call transcriptional noise, but occasionally these transcripts can be used to have some regulatory function within the organism. Okay, so sometimes they're functional in helping the organism regulate its genes elsewhere, but other times they're not. So we've got this problem. How do we distinguish the signal from the noise? Not only that, but sometimes transposons get domesticated, as they say. All right. So one of the classic examples of this is there's a specific species of Drosophila, a fruit fly, where the telomeres, these are the ends of chromosomes that are thought to be important in lifespan. The telomeres are generated by a transposable element just in this one species that copies itself and sends its copies down to the ends of the chromosome. Okay, so they specifically insert in a site that's good for the organism, and that's an example of what's called domestication. So you can have an active transposon in an organism and it's functional for the organism, right? How do you know if it's just doing something for the transposon or for the organism or both? Difficult question empirically. And relatedly, if you just see transposons dispersed throughout a genome, there's going to be some of them that might have organism level functions. Some that are going to be just selected for their position for perhaps replicating themselves. And it becomes a difficult problem to sort out which ones have which functional roles. So the introduction of transposons as a separate level of activity in the genome creates all kinds of epistemic problems for us as these hypothetical scientists. Okay, let's move on now in our little historical survey to the debates in the late 80s and early 90s around the Human Genome Project. And I'm just going to touch on this briefly. Much has been written on this topic. I'm especially interested in the debates that occurred over whether it's a useful and justifiable allocation of funding to sequence the entire human genome. So at this point in time, doing so was going to be very expensive. I think that the estimates were around $3 billion, which was the largest biology project ever funded at the time by the NSF. And there was a certain amount of pushback from research scientists saying, look, that is not a justifiable expenditure of funds. Why not? Well, we think that most of that DNA is junk. It's non-functional. Now, what do we mean by non-functional? Well, we'll get there in a second, okay? But the arguments that were being raised at this time were the same arguments that Ono was raising, right? They were pointing to variability in genome size, and they were pointing to the mutational load argument and saying, look, these are good theoretical reasons, but those theoretical reasons were largely ignored by proponents of the Human Genome Project. Let's look at some of the arguments they presented. So in favor of sequencing the entire genome, did I miss one of these? Okay. A couple of arguments that were presented at the time by Reichsteiner were that there's no general procedure for distinguishing functional DNA from non-functional DNA. Now, look, that's not a very conclusive reason to do it, right? That's exactly what critics were saying. So I'm sure if there was no cost associated with sequencing the whole genome, then that might be a good argument. But the whole point that this argument misses is that this is going to be very expensive. So second, much of this so-called junk DNA could have functions that are as yet unknown. So again, you see this theme, right? That, well, it's probably functional, or much of it could be, and a sort of faith that there's this probably some underlying function there. And then there was another argument I think is kind of interesting that started to arise right at this time, where, look, maybe it's not functional for the organism, but sequencing all of the genome could be really useful for doing evolutionary analyses and anthropological research. So it's not functional for the organism here, but it's more like informative for research scientists. And that argument started getting repeated by many of the advocates in favor of sequencing the entire human genome. So here again, okay, so this is from Paul Berg. DNA replication, chromosome packaging, and segregation. Oh, I'm missing part of my quote there. That's frustrating. I guess what I wanted to highlight here is that he was expanding the notion of functionality in his paper defending the Human Genome Project. One was he's saying, look, these non-coding sequences are essential to life. So here he's kind of alluding to the role that they might play as spacers, perhaps, or generating regulatory transcripts, and saying we shouldn't treat them as junk for that reason. And also, it was interesting how the language starts to slide at this time from junk being now seen as something that's not worth knowing, right? So it's now being considered junk if it doesn't even have some informational value, which is a very different notion. And so you kind of see this slide or shift in the function junk distinction around this time, where functional DNA are sequences that are potentially essential to life or have some informational value. And now junk DNA has neither of those things, right? So it ends up being a much smaller category of DNA, not even useful for information. So now, of course, a lot of these debates flared up again around 2012 when the ENCODE project published its results. And I think one of the missteps in that project was when they sort of advertised to the media that 80% of the human genome is functional. That really created a sort of allergic reaction in many evolutionary biologists who responded with criticisms of that claim, right? That 80% of the human genome is functional. I've written on this topic a bit, and I'm not gonna get into it in too much detail right now because we have other things to talk about. But there's 2 points I guess I want to make around this stage of this sort of historical snapshot. Okay, so one is that the ENCODE project again shifted the function junk distinction. So they shifted it to now they created their own category of biochemically functional. Okay, so they kind of modified the word functional, and they stipulated that something will be deemed biochemically functional if it exhibits at least one of the following properties at least once in at least one of 147 cell types. So A, or 1, it's transcribed into RNA. 2, it contains or is adjacent to a transcription binding factor. Those are the 2 I think that are the most permissive. 3 is methylated by a dinucleotide, is located in an area of open chromatin, or is organized in nucleosomes containing histone modifications. Let's not worry about 3, 4, and 5. Let's just think for a second about 1 and 2. So we already know, as a result of normal co-evolutionary processes between transposons and the organism, that there's going to be a certain amount of methylation happening to silence transposons, but it's often only partial. And there's going to be transcripts that are still generated from these self-replicating entities within the genome. So we're going to have a whole bunch of transcriptional noise, even if some proportion of it's functional. So if your category for functionality is simply being transcribed into RNA, you're now regarding all of those elements that generate transcripts that have been silenced, these inactive transposons. You're now regarding them all as functional. And that's partly what pisses off certain researchers, right? That's just too low a bar. And what it does effectively is ignore the evolutionary processes of going on within the cell. Contains or is adjacent to a transcription binding factor. Let me come back to that one later, perhaps. I think it's best that I come back to that. All right, so here's an issue that, here's an argument against the definition. So transposons, active or inactive, are expected to satisfy many of ENCODE's functional proxies. All right, the majority of human DNA is the result of transposon activity. It's estimated to be about 60%. Some say higher. So most transposons and their remnants are unlikely to make any contribution. Well, I shouldn't say so. This is an assumption. Most transposons and their remnants are unlikely to make any contribution to organismal fitness. They're junk in the 1980s sense of the word. Okay. Now that's debatable, but you know, again, that this is the criticism that's being levied against ENCODE. So the majority of human DNA is not an important research priority for ENCODE. It's junk even in the 1990s sense, because ENCODE is claiming to be interested in, you know, human or genetic elements relevant to human development and disease. And the idea being that, look, when you set your boundaries so low, you're going to be capturing a whole bunch of things that are not in that category. Okay, so I'm going to summarize this historical. Oh, my goodness, sorry. I'm just moving around things on my screen so I can read when I need to read. Okay, so that'll be better. Okay, so here are some historical trends that I'm highlighting. And I want to spend a moment on the first one here, that functional concepts play an investigative role in genomics, this function-chunk distinction. Now, this, I believe, hasn't been adequately appreciated by at least many philosophers, okay? When philosophers are talking about function concepts in genomics, or anywhere, they're usually talking about their explanatory roles, right? So what is the causal role function concept? It explains what an entity does within some containing system to contribute to that system's capacities, right? Explains. And what does the selected effect notion do? It explains why something's there. And I think that those are adequate analyses of how functional concepts play a role in explanation. But what I think gets underappreciated is the function concepts do a different sort of thing when they're playing a role in investigation. And specifically, they mark the boundary between the part, in genomics at least, between the parts of the genome that are deemed worthy of scientific investigation or not. That's what the function-junk distinction is doing. And the boundary of putative functionality has gradually expanded from sequence-specific protein coding regions to anything that's biochemically active. And again, at each of these stages of expansion, we could stop and ask, is this really a useful distinction? I mean, I haven't dug into that as much as I could have, but it's really an important question to be asking. I argued that the last extension was not an intelligent distinction because it just cuts the boundary between what considered functional and not functional. It places too many things in the putatively functional basket. Theoretical considerations about C-value and transposon dynamics are consistently ignored, especially in these last two phases of big biology, right? It just wasn't on the table. People were not taking seriously these theoretical arguments. I have some, hypotheses about why they're doing that. I think that the science of genomics is very experimental and a little bit averse to theoretical arguments in general. I think that many of the people working in this field just aren't trained in evolutionary reasoning, but those are just speculations. All right, so I want to shift now. So what I've done is I've summarized the way that the function-junk distinction is being used over these historical snapshots. We'll come back to it at the end of the talk. But I want to shift slightly now to this methodological problem of causal role myopia. And I've hinted at this already when I've had us imagining what it's like to be a research scientist tasked with the problem of distinguishing a an element that's functional for the organism versus not. Okay. So most biologically active non-coding regions of the genome are functionally ambiguous. Okay, that's due to this problem of gradual functional decay, right? So they keep many of their sort of functional hallmarks long after they're not functional anymore. Due to transposon domestication, selection at multiple levels, and other things that I haven't even gotten into. Okay. So it's just a genuinely difficult problem identifying a truly functional element from one that only has pseudo functionality. When analyzing these regions, it's difficult to rule out the existence of some potential organismal function or other. And we've seen this over history where this argument keeps coming up. Well, maybe there's some organismal function, some cryptic function that we haven't found yet. So let's keep looking for one, right? So meanwhile, and I think this is also an important point that perhaps I haven't defended enough in this talk, but economic incentives and scientific bad habits encourage researchers to keep looking for an organismal function. What do I mean by economic incentives? Well, what I mean here is that you can you could do what the Human Genome Project did and the ENCODE project did and promise to your funders that if you keep mining these regions of non-coding DNA, we're going to cure cancer, and there's probably all kinds of undiscovered functional benefits to doing this. And there's so much of it that we haven't explored. So keep giving us money and we'll keep looking, and you just wait and see how good it's going to be. Now, how long can we keep making that promise, I wonder? What do I mean about scientific bad habits? I'm among those who are frustrated by the fact that the C-value paradox, variability in genome size, and the mutational load argument aren't being taken as seriously as they should when making when deciding when encoders making their claims about functionality, for example, let alone the fact that it's very rare to see any of these researchers actually take seriously the coevolutionary arms races happening within the genome between transposons and organisms. They just aren't employing these theoretical frameworks that seem to be so useful and informative for understanding what's going on within the genome. So alternative hypotheses are systemically overlooked on this sort of framework that predominates. So I've defended this argument in more detail in a paper that I think I have a citation for down there at the bottom, if you're interested, okay? So now I'm gonna move on in the last few minutes here and talk about an alternative approach to looking for genomic functions, or just understanding what's going on in a genome by applying concepts from ecology, and then I'll conclude with an argument, okay? So, all right. So genome level ecology, the basic idea is that we can think of transposable elements as what I'm calling ecological agents. And before you get worried, I only mean agents in a minimal sort of sense here, okay? As something that kind of has interests of its own. Interests in a very non-intentional sense, okay? In that there are certain things that it can do that benefit its survival and reproduction. That's all I'm talking about. So transposable elements move around like organisms in a genomic environment where certain regions, or what we could consider as habitats, are more suitable than others. So here is an example, this diagram of the ALU element in humans. This is just one species of transposable element depicted in green. And you can see here that it is absolutely not randomly assorted throughout the human genome, right? You've got islands and some sort of patchiness happening over here. You've got chromosomes 16 and 17, which are almost entirely ALU, or 19, and some chromosomes that have very little ALU in it, right? So there's a kind of non-random physical distribution of this one specific type of genetic element. There's other interesting things about this element. It actually depends on another transposon called line 1 to engage in its replication. Line 1 generates some enzymes that ALU harnesses to replicate. So there's actually a kind of parasitic relationship between two transposons within the human genome that result in this pattern. And so the basic idea of genome level ecology is that it's possible to apply concepts, models, and methods from whole organism ecology at the level of the genome to explain the distribution and abundance and potentially diversity of transposons. So let me give you a couple examples here. So many of us are familiar with the concept of an ecological niche. So anywhere you go around the world, if you go to the edge of an ocean and you find a little protected pocket like this that periodically gets replenished with nutrients and oxygenated seawater, but is protected from the surge, you're going to find a diversity of organisms that are specialized to inhabit that niche, right? We just know that you're going to find it. And so an interesting question that arises is, when you look at a genome, a eukaryotic genome, could there be niches in the genome that are kind of analogous to this? And one of the interesting ideas around this is that here's an interesting place to look. If you go right up to the edge of a protein coding region, that's a kind of a sweet spot for a transposon to hang out, because if the organism were to methylate that area so close to one of its important genes, it would probably disrupt the function of that gene. Okay, so there are these little places where a transposon can kind of get away with inserting itself that make it more likely to persist there without being silenced by the organism's own silencing machinery. So that would be one place you could look. But, you know, that's a difficult problem. I'm going to talk about other ways that people, including us, have approached this issue of, are there niches for transposons within the genome? So a little while ago, I guess this would have been 2013, a paper came out. Well, let me start here. Okay, so within the discipline of ecology, one of the ways that people look for niches is they basically do it by looking for the absence of niches. All right, what do I mean? Well, there's this theory called ecological neutral theory that makes certain predictions about how you would expect species to be allocated in the landscape if there was no ecological pressure for them to be in one space or another at the species level. Okay, so let me put it differently. If being a fig plant versus being a eucalyptus plant had no difference as to whether you could occupy a particular region, then the system would be ecologically neutral. Okay, if your species membership doesn't matter to where you end up, then the system is ecologically neutral. So that means, what's a niche in this? A niche is a relationship between the properties that are unique to a species and the environment, such that you know, being a member of a certain species makes you better at inhabiting a particular region. Okay, so one way that ecologists ask are there niches in this area is they say, does this area conform with neutrality or not? How neutral is this area? And a little while ago in 2013, some researchers applied that model to the genome. I can't remember which genome they were looking at here, forgive me. Maybe it was a human genome. And they claimed that indeed, neutral models explain the distribution of transposable elements. That would mean no niches, okay? Well, they applied, there's two problems with that. So we responded to this in a paper in molecular ecology saying, not so fast. There's at least two problems, maybe three, with your analysis, and they're really serious problems. One, there's a kind of equivocation that occurs in the notion of neutrality, and I think it's quite interesting. There's two notions of neutrality. There's sort of at-a-level neutrality or inter-level neutrality. So, you know, being a neutral gene is an inter-level neutrality. That means an insertion or mutation that occurs within the genome that affects the organism, right? Neutrality in ecological theory is very different. It just means neutral with respect to your species membership. right? You can either persist here or not, and your species membership doesn't matter, but there can be intense competition happening or whatever. That's a very different notion of neutrality. So we had to disambiguate that. And then we had to point out certain other aspects of the ecological neutral model that just aren't realized in genomes. So it's a bit technical at this point. I don't really know if you want me to get into all the details. Feel free to ask. But our conclusion was that this argument that there are no niches doesn't work. It doesn't work. Okay. So positive evidence, well, we undertook a transect analysis of the cow genome. This was largely Brent Saylor's PhD thesis. So transect analysis is where you go along some linear transect, like up the side of a mountain, and you just document what composition of species exist at every so many intervals of space, and look for correlations often with abiotic factors. So altitude or rainfall or something like that. And do you find systematic changes in species composition and these factors? That's one way of looking for niches. And so we applied that to communities of transposable elements distributed along the linear transects of the cow genome. And indeed, we found that there were correlations between gene density. Certain areas are very gene rich, and it's not the number of transposons you find there, but what you find are there are certain communities of transposons that tend to hang out in the gene-rich areas, and then other communities that hang out in different gene-rich areas. So that's kind of an interesting finding, right? It's not just that transposons avoid the gene-rich areas. It's not quite that simple. It's that there's certain communities that seem to get along in those areas better than others, which is interesting. That's sort of niche looking, right? So there's also some transposon communities that seem to hang out on particularly large chromosomes, and other communities that hang out on relatively small chromosomes as well. So chromosome size seems to have effect on community composition of transposons. So again, is that 100% clear? It's a niche? No, but it's, pretty strong evidence. It's getting there. And what I want to do here is, I hope I have a few more minutes. I've got to stop soon, I know, is hand wave a bit and say, look, you can imagine research continuing like this, just as it does in ecology, where you continue to refine and refine, and eventually you understand the specific causal relationship between a species and a particular feature of the environment, or in this case, a particular transposon type and some feature of the genome. This looks promising. All right, beavers. Ecosystem, this is the last example, and I'm almost done. Ecosystem engineering. So that's a concept from ecology, and what we see at a very macroscopic scale is that, when beavers are present, they can have these sorts of these effects on the landscape. And is it possible that there's something going on with transposable elements in the genome where they're having a kind of effect? Well, this was largely the work of Stefan Kramer, my colleague also at University of Guelph. We created a simulation model where a transposon element is released into a prokaryotic genome. And in the model, we could vary all kinds of parameters and set them at either really high or really low. How much junk is in this genome? What is the rate at which that transposon reinserts itself? How likely, if it inserts into a gene, will it cause the organism to die? And so we ran a whole bunch of simulations. And in most of the simulations, 90-something% of them, one of two things happens. Either the population crashes because the transposons, it's basically a mutational load problem. The transposons just, they kill the organism, okay? And that happens often. Or there's strong selection that purges the genome entirely of transposable elements. They just disappear. But there's a handful of cases, roughly 8%, where the two persist indefinitely. And these genomes get massive, these simulated genomes. They just keep growing and growing and growing. And so we were like, why in these cases doesn't either the population die or the transposons get purged? And it was tricky to figure out because there wasn't any one parameter setting that resulted in that. There were these different combinations of parameter settings where you'd get that outcome, okay? And so eventually, when we looked at it, we found that actually in all of those different parameter settings, there was a common thing, that there was a stable TE density there's a stable density of TEs that are active versus non-active. Okay, so that's ones that can continue to replicate versus ones that are essentially dead on arrival. And the only way to establish a stable density like that is for a TE to generate a certain proportion of inactive copies for every one active copy. Okay, so I'm a TE, I want to persist in a genome. I'm going to create 5 active copies that I can reinsert into for every one that's going to go on copying or something like that. I don't know if the number's 5. Okay, so that looks like that's just an outcome of this, this simulation. And we make an argument that that's a case of ecosystem engineering effectively. So, again, what I want to do is hand wave at this point. If you wanted to find out if that's going on in the genome, what you might do is create more simulations and figure out what the sort of characteristic patterns of this phenomenon are. There's presumably going to be sort of certain conditions under which this can occur and can't, right? And certain sort of hallmarks of an engineering process. Maybe you get it concentrated on one chromosome, right? Maybe it becomes very spatially segregated. I don't know. But the idea would be to find out this process, which we're pretty sure at least happens in our model. If you keep running simulations, you could better understand what that process looks like and then go looking for it in the genome. Okay. All right. So final argument. This is the conclusion. So the function junk distinction is an investigative tool for guiding research efforts towards certain genomic regions and away from others. However, this tool is a blunt instrument, and it's subject to misuse, right? It's blunt in that for various reasons, right? It doesn't take into account gradual decay. There's different things that are getting lumped into each of the 2 categories of either functional or junk at different times. And it's subject to misuse in that it's not very well regulated. I mean, just over time, different people have been able to stipulate what gets to count as junk. Hypotheses about the potential functions of transposon insertions are easy to generate and difficult to falsify. That's the causal role myopia problem. So you can effectively go on forever claiming to look for some putative organism level function and keep getting your funding that way. However, an ecological perspective on the genome identifies patterns of transposon abundance and distribution that one would expect, all things being equal. Right? So for example, in our model, we ran simulations of the model with no organism level benefit at all. Every time a transposon lands in a gene, it either disrupts it or it's neutral. Okay. And still there was persistence of these things. So it gives you an idea. So understanding these ecological processes gives you an idea of what to expect. Therefore, genome level ecology potentially offers a superior framework for distinguishing regions with a likely organismal function from ones generated exclusively by these intragenomic ecological processes, right? So to the extent that you find deviations away from what you would expect just from these ecological processes in the genome, just by transposons doing their thing, to the extent that you get a deviation away from that, you might be able to infer, hey, maybe there's some other process, for example, selection at the level of the organism happening here. And it seems to me that that's a more effective way to go about looking for genomic functions. Okay, that's it. Thanks.
Thank you very much. Very much. That was great. Let me move to maybe you could stop sharing the screen.
Oh, yes.
Thank you very much, so that we can see everyone on the screen. And of course, you can go back to the slides if needed. One thing I wanted to say, in addition to thank you for a great talk, is that we're very lucky to have some of the main people who have written on this question, including Fort Doolittle today. So that, I mean, I really look forward to the discussion. The other thing I wanted to say is, as always, please introduce yourself in just a few seconds to let Stefan know if you're a biologist, a philosopher, because of course the answers often very much are different depending on who you are. So yeah, so who wants to start?
Well, I'll start.
Oh yeah, please go for it.
Because I've already been introduced. I was thinking that Stefan's approach might actually be applied to real ecosystems too. I mean, ecosystems, because I mean, I think I speculated on the evolutionary forces that create transposable elements, but now that they're fixed, or at least those forces are fixed, or at least satisfied, then ecology comes in, right? I mean, and I think probably you could just extrapolate that up to higher and higher levels. That ecological rules dominate at a higher level what evolutionary rules dominate at a lower level. I mean, I think you said that.
Okay, interesting. So there's a part of my thinking on this that I didn't touch on because it gets a little technical and hard to explain, but it has to do with the relationship between ecological processes and evolutionary processes.
Right.
And you and I have talked about this before, but I think that, in a nutshell, it's a matter of which causal processes you want to foreground versus background. And,
sure.
and
right, I think
purely
so.
ecological approach is looking at relationships between properties, typological properties of a species and how they interact with specific features of the environment. And you may not look at evolutionary change in ecology. You may look at a very short-term interaction between a species gets introduced to an area, and is it gonna increase in abundance or peter out? And evolution is a kind of longer-term, often higher-level description where those local ecological processes just kind of get ignored, right? You don't try and get into the weeds on what caused something to be fitter than another in terms of its relationship to the environment. You just say it was fitter, and you make your predictions based on that.
Yeah,
so
exactly.
I think now, if I understand you, you're suggesting that this whole thing can just be, it's all ecology and evolution. It's just all these levels of it goes up higher.
Right.
What I'm describing between the organism or within the genome could be bumped up to continuous levels. And
right. I think that's
yeah,
right.
sure.
Right.
Yeah, why not?
Right. Yeah.
One quick question to Ford. What did you think about the historical account of Stefan? I mean, I just want to know if you thought it was faithful.
Oh no, it was very good, very good.
Papers.
It was very good as far as I can remember those things. Yeah, I mean, it was 50 years ago now, practically. But yes, no, I think the memory is very good, and the historical reconstruction is very good.
Okay, excellent. I have a quick question, and then I will let others ask questions. I mean, I have two, but maybe I'd start with that. So let's assume that this ecological perspective is useful, and it sounds like it could be very useful. You were not entirely clear about the meaning of functional in this sense. So in a way, I was expecting a little bit more. I know you rushed a little bit at the end, but when you said it is, there is a sort of organismal functional perspective here, which is renewed by this ecological approach. I was expecting a little bit more about the exact meaning of functional in that sense. Can you say a little bit more about that?
Yeah. Okay. Yeah. Thanks. That's probably quite important given the sort of slippery notion of functionality in genomics. Okay, the slipperiness of the notion. So I guess that there's a sort of common thread to what gets counted as functional in genomics over time. There's sort of is it doing something important for the organism? Kind of, okay? What do you mean by doing something important? Well, we could debate about that, and I've got some positions on that, but at the very least, does it make a causal role contribution to the systems in the organism that produce the organism's capacity to survive and reproduce? Okay, so when I say is a particular genetic element functional, that's what I mean. Does it have a causal role in allowing the organism to persist or not, right? And my thinking here is that if you jump down to the level of ecology within the genome, now you might end up talking about functional in a different sense when you're down there. And I think that one needs to be very careful with the use of language. Like I would probably avoid the F word when I was down at that level, okay? And I would talk about an ecological role. So does this transposable element have the ecological role of being an ecosystem engineer? And if so, what kinds of patterns would we expect just with that process alone, okay? And then we go, oh, look, it does. We've got species A, B, and C, and it's doing its ecological engineering thing in A, B, and C. But look at species D, same transposable element, and it's doing something totally different. Now I go, that's a kind of prima facie reason to think it might have some functional role in species D. So I'm going to stick with functional role for that, partly because it's sort of what's been kind of common within this field of genomics. And you can, there's always a danger in inventing any new term, right? I mean, there's been so much confusion already. So that I think is, if I'm going to try and be terminologically precise, which I realize I probably wasn't being, that's what I would do. I would say, when I'm at the level of the genome, talk about ecological roles. Don't have to use the word function, I don't think. But be precise about them, right? Is it in a parasitic relationship with another transposon? If so, what does that mean? Is it is there a diversity stability relationship at the community level of transposons? Something like that, right?
Yeah.
Yeah.
Yeah, I really like that. But one worry is that the people who fight over junk DNA might think that this is a little bit orthogonal to their views about what is junk, non-junk. I don't think it is. I'm just asking
I
you
see that
to
as
what
a
extent.
benefit. Yeah. I'd like to sidestep the debate because I haven't seen it as being a particularly productive debate. I think that in the first two historical snapshots, it was somewhat productive to say, look, there is this category of DNA that's not functional in this sense of having a sequence-specific function for the organism, a sequence-specific causal role or selected effect, actually, in that case, for the organism. And perhaps it's a lot of the genome that's like that. And then there was a kind of expansion of what might get considered as junk. Well, that also includes selfish DNA, and now we could probably identify a lot of other players that could go in that category. So there was a kind of utility that the distinction had, but the greater that, the more the larger the basket of junk gets, and the more diverse the things are that fall into that basket, I guess the less useful the distinction is, right? So what are people arguing about now? I'm not even sure they're clear on what they're arguing about. I mean, you see people using the phrase long thought to be junk, now considered functional, and they aren't always identifying a sequence-specific function. Sometimes they'll just be identifying like a spacer type function. Well, we've always thought those were there. Long thought to be junk, now considered functional. Well, not in that sense. So I think that the function junk debate is partly plagued by an unclarity about the use of these terms. And so finding things that are orthogonal to it is promising.
I like that. Thank you very much for a cool answer. And I see that Pierre-Luc is clapping. Pierre-Luc, you can intervene if you want. Fridolin, you're next in line.
Hello, I'm Fridolin. I'm a philosopher of biology, I think. I really like the talk and the ecological perspective on the genome, but I will come back. I'm sorry. I will come back to the debate. And so you talked about functional decay, and you talked about you questioned whether we can really talk about junk or non-junk, but we should talk about levels of junkiness. And so doesn't that imply also that we cannot really talk about function as a binary category? And it doesn't make sense to ask the question, what percentage of the genome is functional, I mean, it's the same as if we ask, like, what proportion of people are tall, right? Something like that. So could you imagine a way of rephrasing this question and asking a different kind of question? I mean, I don't know, or like making a
well,
continuous...
let me try this out. Okay.
Yeah.
Thanks for the great question. I have to think about it here. Okay. So if we pin the notion of function to having some sequence specific selected effect on the organism. Okay, so that's sort of, I would think, the Ono notion and arguably the 1980s notion. I think you could say, it's a meaningful question, though. It's a meaningful question, though epistemically very difficult to answer. How much of the genome is functional? You could go nucleotide by nucleotide. You'd probably want, how would you do it? God damn, it's hard to think. All right, I want to just say, it seems not so implausible to me that if you draw the distinction that way, you could say definitively that there's some amount of functional DNA in this organism, and that one's got more and that one's got less, because you're talking about something very specific here. Part of the problem with junkiness is it's more of a kind of matter of degree. You're getting more and more and more junky over time. There's a kind of decay rate or increase in junkiness. And I'm not sure that there's an increase in functionality. I'd have to think about that. I think that's the real question that you're answering. Can functionality come and grow as a matter of degree in the genome? Good question. I don't know.
Aren't they just two sides of the same coin?
I don't think they are. No, I think that it's not quite as simple as that. At least it depends, right? If we anchor on those original definitions where function meant having a sequence-specific selected effect, but junk meant not having it. There's sort of more ways to not have it than to have it, right? Not having it is a gradual notion, whereas I think having it is more of a, you have it, maybe you're right. It's just maybe it's just a logical, you're losing functionality gradually. Yeah, no, okay. I think it's a tough question. I've never thought about it before. So again, I'm thanking you as I think about the answer. I'm going to have to think about it. Maybe it's the case that, functionality decays gradually, and it follows logically that as you become more junky, you become less functional. I need to think about it.
Okay, fair enough. Thank you.
Yeah.
All right. Alok has a question.
Yeah. Stefan, your research program is amazing. Thank you. The historical platform, I'll introduce myself in a bit, but the historical platform you set up in the first part of the talk with the 4 epochs allows us to participate in your research program. So thank you. My personal background begins with protein biophysics and protein engineering. I will tell you a small story that's relevant from there. And then through biotechnology and other things, I now do history and philosophy of science with, I study bioengineering labs as well as developmental biology labs. So there's a parallel situation to the function junk story, and functional assignments turn out to be junky, and junk assignments turn out to have functions that were not in your catalog, right? There's always this persistent situation in biological research that there is more natural history to be had. And the incompletely done natural history will disrupt your theory. So in protein science for a long time, the impression was that every amino acid in a protein sequence played an important role for an enzyme. Like the classic thing was hemoglobin, the sickle cell anemia, one amino acid change destroyed the hemoglobin's function, created disease. So every amino acid would be like that. Now in the 1980s, when you had the ability to make DNA oligos for the first time, you could design engineering are these junky words, but they do allow us to talk about experiments. So in the 80s, for key proteins, people would replace every location in the sequence with all the other 19 amino acids.
Mm-hmm.
Right? And it turned out 99% of the sequence could be replaced one at a time with no effect on the functioning of the protein. It was done with lambda repressor, lac repressor, right? So the function density on the protein structure with respect to sequence turned out to be very weak. It allowed people to say, let's cut out all the other junk sequence and only make a 16 amino acid peptide with the catalytic protease enzyme site. Doesn't work, right? So it's not exactly junk. The scaffold is not exactly junk. So in the last 3 slides, when you developed and I want to kind of rephrase one of your phrases, coevolution arms race, let's call it coevolution structuring race between the ecological niche and the organism, right? If there is a coevolutionary, co-constructive structuring race, so the 60 amino acids for lambda repressor, right? You cannot delete them, even though each of those sites is tolerant of 19, you replace the amino acid with the other 19 or 16 amino acids, right? So you have that.
Yeah.
Okay, so this is just my introduction to myself. Now, one of the things that, like one of the epochs I would add is, Craig Venter building the genomic age with ESTs, right? Express sequence tags. This is the business part of the genome. He built his whole career. And then in 2018, produces the minimal organism, aggregating all of human knowledge of what would be the minimal gene functions you need. And it turns out that they needed 63. They only got 1/3 of the genes necessary to produce a viable organism. It was a beautiful audit of humanity's knowledge of which were the functional genes. The Minimal Organism Project failed in that sense. He needed to put back. And so it's again a story that's similar to the problems you're constructing. I want to burrow into some things you began in the beginning and then to the end. You use these 2 phrases, the transposon structuring the genome and structuring of the organism. Now, these 2 phrases, structuring the organism or structuring the genome, are at the other end of information, organizations, and structure, right? You're using structuring almost in the language of physiology.
Okay. Yeah, I don't think I was very self-conscious about my use of that word. So you may be drawing my attention to something that I was not aware of.
What is valuable, what is terribly valuable about it is that in that phrasing, you're no longer seeking a program, a mechanistic program that will do the information to structure process.
Right. Yes.
It's very
that's one of the things
interactional.
that, So sometimes when I'm trying to sell this package of ideas, I contrast it with a view of the genome, which is the sort of the familiar view of the genome as something that's a recipe or a blueprint. Because I think that's part of it's a feature that leads to the causal rule myopia thing, right? That, well, it's a part of that. You approach it asking yes or no, is it part of the blueprint? And it's very hard to show it's definitely not, right? And so if you end up with these, well, here are the baseline processes that happen, and some of them result in blueprint components and some don't. I think you end up from a more sort of You spin your wheel. It might just end up being very hard, but my hope is that you spin your wheels less and avoid these debates. So that might be what you're touching on there, right? Like, here's how, and you can do it with simulations. You can say, we got this transposable element, we got these genes, here are the range of structures that we arrived at with no organism level benefit of insertion, right? Now add organism level benefit of insertion and see if you get different sorts of genomic structures. How do they differ, right? You can totally do this kind of bottom up reconstruction, in theory at least.
I have one last comment. So you in your second epoch in the 4, there was Leslie Orgel, right, who did a bunch of work on DNA synthesis, and his stream of work and the future people, they produced in vitro evolution, right? So the RNA world, Shostak. So one of the interesting things about bioengineering is it relates to your last 4 slides, that in certain ecological niche, that ecological niches can have neutral presences that are co-structuring, right? So much of bioengineering is making evolution happen in a Petri plate. It's not intentional design as much as constrained evolution towards, it's exactly what you're describing in your last 4 slides. Whether it's Francis Arnold, whether it's Jack Shostak, whether it's David Baker doing protein engineering, all of them are not doing engineering the way mechanical engineers do. They're growing a platform of genomes and organisms into particular properties or capacities. It's your last 4 slides. So
yeah, interesting.
Thank you.
Okay, I should learn more about genomic engineering in particular, I guess. Thank you. That's useful.
Yeah, that was very useful. I don't see any other questions. If someone has a pressing and relatively short question, it's, oh, there's a comment by Paul.
Let me open my comments.
Paul, do you want to say more on that? Or that's just what you want? You said it.
Right. Yes. Now I remember that paper, which I should reread.
Well, the message then is that it should be in your Cambridge element, if I understand correctly. It should
I'll put it in for sure. Yes.
And well, yeah, I think we all look forward to seeing this Cambridge element.
Yeah.
This sounds very exciting. So if there is no other question, which I think I can confirm now, I really want to thank you again, Stefan. That was great. And I really enjoyed the discussion as well. So thank you so much.
Thank you very much. Okay, take care.
Bye. Take care.
Bye-bye now.